So the last time that we met, we talked about the parts of the heart, and we were going over the blood vessels of the heart, arteries and veins. And we were also looking at the heart externally, in which we were naming some of the areas where we're going to find certain things. Now, if you look down at the bottom part of the picture at the bottom, you can see that we've got a coronal section of the heart, right? Which is exactly what we're going to do in lab. We're going to take a heart, and so we need to be studying these pictures, so when we get to an actual sheep heart, it'll be familiar to us. But we're going to take it and we're going to do a coronal section of the heart, and we're going to look inside. And we're going to look for things like the atria, the ventricles, and you're paying attention to how they're different. Like, is the right side and the left side different, and why would it be different? We're also going to be looking at valves. We'll have 4 that we'll be thinking about, and they prevent the backflow of blood. So we've got to think about why we have valves and what they're doing. And so looking at this picture that we see, first of all, we can see the atria from this coronal cross-section. So here, this is the right atria here. And basically, that is where blood is emptied into the heart from the superior and inferior vena cava. So the superior and inferior vena cava is a gigantic vein that is dropping off deoxygenated blood to the right atria. Everybody see that? And so basically, once it enters the right atria, this chamber fills up with blood. And then when it actually releases the blood into the right ventricle here, okay? And so this is the ventricle. And what you see is from the right ventricle, or actually let's look between the left and the right ventricle, there's a little valve in between them called the tricuspid valve. So, and that just means it's a 3-flap valve. And it's important to remember that because there's a tricuspid on the right side and a bicuspid on the left side. So the tricuspid is found on the right side, and it separates the right atrium from the right ventricle. So then, once blood fills into this ventricle, that tricuspid valve is going to close and prevent the backflow of blood back into the right atria. And it does this, the tricuspid valve does this because it's actually connected to these little tendon-like structures here that you see. They're called chordae tendineae. The chordae tendineae attach to actual muscle. So there's little papillary muscles right here attaching the chordae tendineae, and when that muscle contracts, that's helping that to pull down that flap, and when it releases, the flap closes up and blood cannot backflow into the right atrium. Then that's closed off, so the only way, when that ventricle pulses, and we'll talk about the whole contraction part of the heart a little bit later, blood is leaving the right ventricle and it's going to go through the pulmonary valve. We call this valve the pulmonary valve because it's part of the pulmonary circuit, right? It's taking blood and sending it into the pulmonary circuit. So it's called the pulmonary valve. So the blood leaves the right ventricle and goes to the pulmonary veins. And it's going to the pulmonary veins through the pulmonary valve. And what was special about the pulmonary veins? I'm sorry, I messed you up. I did. I said it's the pulmonary arteries. What is leaving through the pulmonary arteries, through the pulmonary valve to the pulmonary arteries. Slip of the tongue. So it's an anomaly to what you think. So we define an artery and a vein. We have the definition, and that is artery away from the heart, vein towards, right? A lot of times we like to think about what state the blood is in, depending upon the gases, right? But this is the exception to the rule. And so you always have to remember the pulmonary artery and the pulmonary vein don't behave like the others, in that the pulmonary artery is carrying deoxygenated blood. And this can be very confusing to students. This is why I'm pointing this out, because most of the time they're colored red and blue, right? And we grow to make that connection that red means oxygenated and blue means deoxygenated. But that's not the case when we're dealing with the pulmonary artery and vein. So I make a big deal to point that out to you so that you don't get confused. So the pulmonary artery is, by definition, taking blood away from the heart. And where is it taking blood to? The right and left lung. Remember, the heart is housed in the mediastinum, in the pericardial cavity, and blood will be pumped from the heart through the pulmonary arteries to the lungs, where it will become deoxygenated. And when we get to the respiratory system, you'll really get, what did I say? It will become oxygenated. It's going to the lungs so that it can become oxygenated. And when you actually get to the respiratory system, it'll make a lot of sense on how that happens. Because when it gets to the lungs, you're going to see all these capillary beds in which these little bitty capillaries are having this exchange. Gas exchange occurs. And these same little capillaries are found all throughout the body. Our bodies are full of capillary beds where gas exchange occurs. And here's another thing that can be confusing, if I can not confuse you any further. And that is that all of the oxygen in your blood is never 100% completely dropped off unless you have like an issue, right? So your blood is actually always carrying some oxygen, a percentage of oxygen, in case and it's like a reserve in case you need that oxygen. So even though it's easy to think of in terms of oxygenated and deoxygenated, it's actually more oxygenated. Yes, it's actually more appropriate terminology to say oxygen-rich and oxygen-poor. But it's easier to think in terms of red and blue, right? So that's kind of the reason why we simplify things. But always remember, blood never 100% drops all that off. And remember the little hemoglobin molecule from the last chapter? Each little hemoglobin molecule could carry up to 4 molecules of oxygen. And remember what metal was found in the molecule that bound? Iron. So that's why you need iron. If your iron's low, you're going to feel fatigued because you're not getting enough oxygen. All right. So the pulmonary arteries are special. And if you look on our actual picture here that you're looking at, the pulmonary arteries are actually blue because of the deoxygenation. And that's what it's showing you, is that it can be very confusing to students because we're dealing with an artery, and typically what do you see an artery to be the color of? Red. And that's why that one's blue. So it goes off to the lungs, it gets the oxygen. Where does it come back in? Both, yeah, left and right pulmonary veins. So, yes, and so the veins here, these are them, and here's where they enter. All right, and this one actually you can't see because it's where it's located. You can't see the whole thing, but it also is entering in over here. These that you're seeing are entering in here in the left atria. Y'all see that? So, and they're red, but they're veins. And it, again, it's because they're going to the heart. But now they're red because why? They're pumping oxygenated blood back into the body, oxygen-rich blood. So as everything enters into the right atria, then you have contraction, and that blood will move into the right ventricle. And what do you notice about the right ventricle? I'm sorry, the left ventricle. The right ventricle has less muscle than the left ventricle, right? So all the blood's coming in the left ventricle, which has a really thick muscular wall, because it has a long ways to go. The lungs wasn't very far away. Didn't need quite as much muscle. But now we got to have a lot of muscle in the left ventricle, because when blood leaves the left ventricle, it's going out the aorta. And when it goes out the aorta, and you can see, let me get some of these marks off so to be less confusing. So when it goes out the aorta, the left ventricle, and enters into the aorta, it goes through the aortic valve. So the aortic valve and pulmonary valve are pretty easily to remember because they're named for the structure that they're entering. But you got to remember the tricuspid and bicuspid valves, and they're named according to flap, and tri is on the right, bi is on the left. Okay? So once the blood leaves the left ventricle through the aortic valve and through the aorta actually, and you can't see this from this picture, but it loops down this way. Y'all see that? All the way down to hit the lower extremities and all the digestive organs and all that body. And it has these little branching arteries coming off the brachiocephalic trunk to feed, the head and the arms, the oxygen-rich blood. So you can see all of that pretty easy. Any questions about that? They're missing a part of their heart? Yes. There's actually a little valve, or it becomes a, it goes away at birth when they take their first breath, but there's a little hole in between the right and left atria where blood mixes. Yeah. We'll talk a little bit about that development process, but yes, it is very different. All right, so here's the valves we've been talking about. We pretty much talked about all of these valves. Now, we also, this right and left, I told you the tricuspid and the bicuspid. Let's learn all the terminology right quick. So for the valves we were talking about, the bicuspid is also known as the mitral valve. So that's another word for bicuspid valve. And these three valves are known as your atrioventricular valves. Basically, that just means they're the valves that separate the atrium and the ventricles, right? And the valves that we talked about that were pulmonary and aortic, those are sometimes called the semilunar valves. And so basically, what are valves? They prevent the backflow of blood, and you can actually see that the atrioventricular valves prevent backflow of blood from the ventricles back into the atria. And you can see that the semilunar valves prevent the backflow of blood from the back into the ventricles. Okay? So at this point, could you tell me the flow of blood through the heart? So blood enters, let me just go through it with you right quick, because this is something you will have to do in lab and lecture. You need to know and be able to tell me the flow of blood, okay, through the heart. So blood enters, and at any point, what if I start anywhere I want to start, you should be able to go through it. But just start making it easy, starting at the superior and inferior vena cava. Blood enters through the superior and inferior vena cava, entering into the right atria. It's in the book, and it's in your notes. The right atria, blood leaves the right atrium and goes to the right ventricle, going through the tricuspid valve. Then the blood leaves the right ventricle. It leaves the heart through the what? Pulmonary arteries, and it's separated by, or which valve prevents backflow? The pulmonary valve. It then enters into the right and left lungs, where it receives more oxygen, becomes oxygen-rich. Then it will return to the heart through the pulmonary veins, now containing oxygen-rich blood, in which it will enter the left atrium. From the left atrium, it will enter the highly muscular left ventricle, going through the mitral or bicuspid valve, and then it will leave the left ventricle, going through the aortic valve, and will go out the aorta. Okay, in the aorta, you can As you can see, it loops back. And you've got, the aorta has branches, brachiocephalic branch, subclavian, carotid branches, in which that feed the head, neck, and upper extremities. And there's that pathway. Next slide. The pulmonary trunk, it's just a trunk of those blood vessels. You see how the trunk splits? So it's basically, you know, that it's like a tree trunk. Mm-hmm. I just basically want you to know it. And, you know, when I ask you questions, it's really not terminology specific as much as, I mean, you need to know all the terminology that we talk about. Mm-hmm. If I know what you're talking about, I'm happy. And the thing about it is, it's going to be a multiple choice question. So, well, they're the same thing. The pulmonary trunk is branching into two smaller arteries, but the pulmonary trunk is part of the artery. Which what? Yeah. Yeah. Well, the organ it's going to is the lungs. Right. The left and the right artery, pulmonary artery, would be easy to identify. You see what I'm saying? Because that's not here. One's going specifically left, one's going specifically right. So you should be able to denote that. So that basically keeps you from writing down all that, but it just gives you a slide where you can see where it all actually happens. And so I want you to be able to do that at any point. I could pick any point and then say, what next? And I would want you to be able to do that. Okay? Can y'all do, you may not can do that now, but I feel like pretty confidently you'll be able to do it soon. All right, looking at coronary circulation, which we've been looking at the coronary arteries and veins a little bit, and we've kind of been denoting them. And they're separated. So we've got the major coronary arteries at the top and the major cardiac veins at the bottom, right? And so in a picture, they look like they're stacked on top of each other, and usually there's an artery on top of the vein. So you can see this better by looking at them separately, and also the heart is transparent. Do y'all see that? So what is that? That's giving you, by having the heart kind of transparent, you can see the anterior and posterior coronary circulation. So these pictures are kind of neat to kind of look at where you can see that. But the myocardium is very thick, so the blood that's in the chambers is not enough is diffusing into the heart. So it's the heart has to have its own blood supply. Okay? So that's important. So as blood's pumping to the chambers, which is not enough, obviously, to oxygenate the heart, it's also pumping to the coronary circulation and then pumping back through. And so that's what you're seeing here. And you have, which we've shown before, here's your left coronary artery, and it's actually coming from the aorta. So there's and it's providing the necessary gases for the heart, so necessary oxygen there. So, and here's the right coronary artery. So those major two branches coming off, they come from the aorta. The left coronary artery that you see here actually divides into the anterior interventricular artery and the circumflex. So here's the interventricular artery, and there's that circumflex artery that I showed you a few days ago or several days ago. That's that major one that often gets clogged up and causes people to have heart attacks, right? The circumflex. So there that is. So the first thing you think about is left and right coronary artery. All right, and the left one splits into the circumflex and the interventricular artery here. The right one on this side divides into the right marginal artery and the posterior interventricular artery as well. So here's that right coronary artery here, and here's the marginal artery. You can see that little artery coming off, and the interventricular artery as well. An anastomosis, by the way, just so you know that terminology, is a junction of vessels, and you can see one here, and you can see one here. It kind of favors a capillary bed, but that's just basically when you have a whole lot of arteries kind of anastomosing around each other. So that's an anastomosis, and that's an anastomosis. Now looking at the cardiac veins, we've got the great cardiac vein, which we see here, and it comes down the anterior side of the heart. We've got the middle cardiac vein. Also, the coronary sinus is on the posterior side, and it's like it's almost like a reservoir. It's a sinus. That's why it's called a sinus. So it's a reservoir for collecting some of the blood. And here's the small cardiac vein. So you can see that these are the anterior cardiac veins. The middle cardiac vein comes off of that coronary sinus on the posterior side. All right, so what you want to do is just kind of locate those. And these pictures are really good for locating them. Then, once you kind of learn these separately, then you can start looking at them stacked back on each other. But just having that description there of where to find them, what's going on, kind of helps. And that's the way I learn. Like, I like to take things, break them apart, and then put them all back together. All right, let's talk a little bit about muscle. So, last semester we learned a lot about muscle, right? We learned about the fact that this is not going to be a voluntary muscle. Y'all remember the voluntary muscle is what we spent a whole chapter on last semester. The voluntary muscle, we think of skeletal muscle, okay? So this is cardiac muscle is majorly a part of the autonomic nervous system. What do you think of when you think of autonomic? Automatic. So it automatically happens. You don't have to think about your heart to make it beat, right? So there were some similarities between the cells, y'all remember, of the skeletal muscle and the cardiac muscle, but there were also some differences. Anybody remember a big difference on a cardiac muscle that you're like, that's cardiac muscle? Intercalated disc. Both of you said that. Very good. There's these little disc structures that separate each cell, and you always find those discs on cardiac muscle. It's similar in that to skeletal muscle in that it was striated. Y'all remember the striations? The striations were somewhat different, a little bit different, but it does have the striations. These cells also have plasma membranes, just like the skeletal muscles did. And these are separated by the intercalated discs, and that's one of the things that you remember about cardiac muscle. Cardiac muscle is autorhythmic, part of the autonomic nervous system. These fibers contract as a unit, and it has a longer absolute refractory period. If you'll recall, if you'll remember, we talked about refractory periods in skeletal muscle and also in the nervous system. Y'all remember that? So basically, it's a period of time in which it's not going to fire again. Y'all remember that? So it's a little bit longer. Now, what's really neat about the heart is it's its own electrical impulse, right? You've heard of the pacemaker? So that's going to be your SA node. And I've actually heard or read books call it a spark plug because it keeps keeps it firing, keeps the rhythm going. So basically, you've got a place in the heart called the SA node found right here. And it follows what we call an internodal pathway. So this effect is very similar to a nerve impulse in that it's like a domino effect. Y'all remember the domino effect from the nerve impulse? So meaning that the exchange of ions happens continuously like dominoes. And depolarization, do you guys remember depolarization is that exchanging of ions? I don't know, if you didn't have me for AMP1, that might not be something you remember. But I can see my students going, oh yeah, I remember about that. Do y'all remember about depolarization? Well, it's something that's kind of important because depolarization, we talk about depolarization events all throughout anatomy and physiology. And basically that's how a nerve fires, is through positive charge on the what side of the cell? Outside. And the sodium ion caused that positive charge. On the inside it was negatively charged. So this difference in charge has caused the cell to be what we call polarized. That means it's ready to fire. When it fires, these gates open up and all these positive charges come in, and as it moves down, these gates open up to let the negative charges in, they swap, and then to reestablish it, we have a little pump that puts everything back to where it needs to go. And that was a depolarization event. When these gates start opening up, it's a charge, it's an impulse moving through the actual nerve, yes. The sodium-potassium pump is the actual pump that pumps to the reestablishment part of this. Sodium-potassium pump is a common theme in biology. Like throughout the body, we have lots of them throughout the body, but that is a particular instance of when you have a sodium-potassium pump reestablishing the polarized membrane. And that's what an impulse is the exchanging of those ions creates energy for it to move. And basically we start at the SA node, and it's like a domino effect. So you'll see here, it actually jumps to what we call the AV node, atrioventricular node, or bundle. And this is what's connecting the atria and the ventricles. So then it moves down at the septum, y'all see that? And these fibers actually split. And they're called the bundle branches. The bundle branches split so that they can serve both sides of the chambers of the heart. So you can see it actually travels down this way till it kind of gets towards the apex. Here we have these fibers called Purkinje fibers, and it actually travels through here and comes out this way. So it follows that track. Now, in the actual heart, you begin with, for the rhythmic chain of events that are going to happen in the heart, you begin with the pacemaker potential. And this is when sodium channels open up, just like what we've talked about in a nerve impulse, and they enter into the cell. The potassium channels close. Okay, so you got sodium flooding in. Then you have the depolarization event where calcium influxes in. The repolarization event, repolarization is establishing back the potential, the membrane potential. The calcium influx ends, and then those potassium channels open up and potassium leaves. The difference between these events and the nerve impulse events is the ion calcium. Calcium is serving more so the role that sodium did in the impulse when we studied that. But calcium is also a positively charged ion, like sodium. Now, the potassium flooding out, remember potassium is also a positively charged ion, so it is not the one causing the internal membrane to be negative. Does anybody remember what causes the internal membrane to be negative? Negatively charged protein anions. Okay? There's just a whole bunch of those, and they can't go in and out of the membrane because those proteins are big, right? Remember, proteins are big and they don't just jump through membranes. They have to have very special transporters or events to occur, like bridges or something to open up for them to be able to get in and out of the cell. But ions can move relatively freely depending on circumstances and channels, right? So that's the events of the rhythmic chain of events for basically the heart contraction. Now you have the autorhythmic cells that we were talking about. You've got a patch of them at the SA node, and I pointed that out here. Here's the SA node here, and then here's where you have the AV node, also known as the bundle of His. That's another name for the AV node. Then you have your bundle branches here, and then here's where the Purkinje fibers are located. So that, basically that impulse transmits in that direction. Now, why is that important? Why is it important to understand that transmission and that impulse? Because it's diagnostic. And when there are issues with the heart, if you can use how it's pumping or contracting to figure out any type of disease issues going on or homeostatic imbalances. So you may go to a doctor and have something going on with your heart, and they're going to order an ECG, which is an electrocardiogram. Sometimes people refer to it as an EKG, but the proper terminology would be ECG. When you go to get an ECG, the doctor is basically looking for 3 major sections on that ECG, and these sections are important by voltage and time, okay? And so the voltage being put out is going to show you, is going to give you a length of how much voltage in millivolts is coming out, and then the time it takes for that to occur is also going to help a doctor figure out how things are going with the heart. And so the 3 parts of that are called the P wave, the QRS complex, and the T wave. So the doctor is going to look at those 3 portions of an ECG to be able to monitor the heart, so see what's going on with the heart. What is happening during the P wave is known as atrial depolarization. So that's when the depolarization, that wave is symbolic of the depolarization of the atria. So you see it on both sides here, right? And remember, those atria contract at the same time. The right and the left are contracting at the same time, right? And the right and left ventricle are contracting at the same time. So blood is not only being pumped through the body at a certain point, but it's also being pumped to the lungs. So those are contracting at once. And the ventricle contract at once. So here's the SA node sending out the message for that to occur. So this, what you see in yellow, highlighted in yellow at the very first picture, is atrial depolarization. The wave that denotes that is the P wave, and this is a normal P wave. Now you can't see, if you were a doctor, you would need a graph behind the P wave to see if that was normal. I can't tell by looking at that it's perfectly normal, because I would need to see the number of millivolts by looking at the height of that wave, and also looking at the number of seconds it takes for that wave to occur by measuring from this point to this point. The next event of importance that we're talking about being the QRS complex is right here, QRS. Everybody see that? Do you see the flat line though that we see here? This in gray? That flat line is denoting the completion of the event. And that's easy to denote because nothing's happening, right? Flat line. So we see the flat line here. So the atrial depolarization is complete, and there's a slight delay between the SA node and the AV node. And that's why you see that flat line in gray in between the P wave and the QRS complex. Now here we are on number 3, we're at the QRS complex. So you've got the Q dip, this R spike, and an S dip that make up that complex. This particular event is known as ventricular depolarization. But here's what you need to know. This event is also part of atrial repolarization. So the atrial repolarization, but that event is totally masked by the event of ventricular depolarization. So what we are measuring here and what this represents is ventricular depolarization. But it is important to understand that atrial repolarization is occurring at this point. And what is repolarization? Repolarization. Reestablishing those ions so it'll be a polarized membrane so another event can occur. So that's happening here. And so now you can see that's denoted in red, right? Yes, what's your question? Together. I'll talk about that later, but it's left and right together. top, left and right together, bottom, atria and ventricles. So now the in red is denoted as repolarization, but you still have the yellow showing depolarization. And so the ventricles are depolarizing at this point. It actually begins at the apex and causes what we see here, the QRS complex. All right. Now, ventricular depolarization is complete. We see another flat line. Y'all see that? Did you have a question? Well, if you get an abnormal, you have to know that. Like there are tons of things that you would know. I have an activity that I'll show you guys in lab, and it shows you the electrocardiogram with the graph behind it. And so if you're a physician, you look at that graph and you're looking for it to cover so many little squares. That's going to tell you the millivolts, and you're also looking for it to span so many squares this way, and that's telling you the time it takes for it to occur. I'll give you an example that I can't remember the specifics because I don't know how to read these. Okay? Well, I mean, but a doctor or a physician would be able to tell you what that indicates. But they could specifically tell you why that occurs. And the thing about it is, for example, some part of the heart may be enlarged, for example, and that might show up on an ECG. And it would be something that, sometimes it'll be something they can treat or some type of problem. It might show that there's heart damage. Some portions of the heart muscle have died and it can't pump the same. So we're basically measuring these pumps, okay, by looking at these waves. So yes, you can diagnose certain things, and I believe there's some pictures in your book that shows, for example, if you get a little squiggly wave, the SA node, the pacemaker, for example, that's how a doctor will say, oh, we need to go in and surgically implant an SA node. That's what they call a pacemaker. Have you ever heard of a individual, if you sit by them, you hear a ticking noise? Yep. Sometimes, and I know that my husband's grandmother has a pacemaker, and when I sit by her I can hear the ticking. And it sounds like a watch, but it's in her chest, and it's that pacemaker constantly clicking. And it's, in order for a doctor, I'm sure they run many tests. Like, you have to recognize in the field of medicine, there's not usually just a one test. And that's why it gets so confusing to people, because they're like, well, this one test says this is what it is. Well, it's not always that. Usually that one test leads the doctor to run some different tests. So they're trying to narrow down what the issue is. So you can use an ECG in conjunction with other tests to come up with a diagnosis. But if you need a pacemaker, that would be something that an ECG would give a red flag for. Hey, there's some wild things going on. And there's pictures in your actual textbook that show you some actual diagnostic pictures. Yes. Stent. A stent is when they go in. They can actually go in and put a little balloon in there and actually blow it up, and it pushes the plaque out of the artery so that more blood flow can go through. So it's basically a procedure. They can also take arteries from other places and surgically implant them in the heart and bypass, like if you had a heart attack and it's because that circumflex artery has clogged up, they can circumvent that by putting another artery in. So there's different things that they can do to help with that. All right, and so here we're right here, we saw ventricular depolarization complete. And here at the T wave, we have ventricular repolarization. So that little wave is showing the repolarization of the ventricle so that another event can occur. And then here's a flat line right after the T showing you that event is complete. So every time you see a flat line, it's a pause, pretty much. That's done. We got a little pause there. No, because the way that I understand this, there's no overshoot. Okay? And to have a refractory period means it's going to take a greater impulse, or greater, and this is like automatic, rhythmic. It's the same. So it's going to be different. All right, so the love dub sound, right? That's what I was just asked about. And this is a sound, and that sound that you're hearing are valves. They're closing. So when you go to take a stethoscope and put it on your chest and you hear that noise, it's the closing of valves. The first sound that you hear are the atrioventricular valves closing. Remember, they close at the same time, right? Because the left and the right sides of the heart are doing their thing at the same time. You just saw that from that ECG. So the first sound is the AV valves closing. So what do you think the second sound is? The semilunar valves closing. Okay, so it's just a swishing noise, and it's denoting those valves closing. I think that might indicate a hole in the valve, and that's a heart murmur. So if you hear an extra noise, it's typically going to be some backflow that you're hearing from that valves could have a hole in it or something to that nature. And at the point at which the atrioventricular valves close, they're closing because the pressures are equal. So the pressure in the ventricles the ventricle pressure is great enough that it's causing the valves to close. So the pressure is a little greater in the ventricles, closing that valve up than it is in the atria. All right, let's talk about systole and diastole. And systole and diastole are the values that you actually get when you take a blood pressure reading. Anybody in here know how to take a blood pressure reading? Do you guys have to learn that at all in your program? You can take blood pressure on the wrist, but I'm talking about the values. I mean, there's blood pressure cuffs that go on the wrist, and you'll get two values, and those values are the diastole and the systole value. Yes. So systole is contraction, and diastole is relaxation. And you'll remember, that's kind of easy to remember because it gets really, really tight on your arm, and then it slowly relaxes. So there's a point at which that's the relaxation point. So you get a systole value on top, as she was saying, and a diastolic value on the bottom. So the steps here are ventricular filling that happens in the mid to late diastole. Blood is passively returning to the heart. At this point, your atrioventricular valves are open. Semilunar valves are closed, so it's passively entering into the ventricles. Atrial systole finishes pushing blood into that ventricle. P-wave and the beginning of the QRS complex is at this point. Okay? So that's in ventricular filling. So you're putting together now those events that we just talked about. What is all happening at what point? We just talked about the ECG. We talked about what the PQRST meant. We talked about the fact that was repolarization and depolarization. Well, what is happening to the actual heart during these events? So when the P wave has ended and we're beginning that QRS complex, here we are, blood moving from the atria to the ventricles. Ventricular systole or an atrial diastole happens, you have a rapid rise in your ventricular pressure. What does that high pressure do? Causes the what to close? All right, so the atrioventricular valve's open, but what's it forcing open? What valves is it forcing open? The semilunar valves for it to leave. It's getting ready to leave at this point. And that QRS complex is complete here. All right. Then we have what we call isovolumetric relaxation, and that's early diastole. The ventricles then relax. The blood is leaving, so the semilunar valves close, and that's where we have our T wave. So now you can put the events of repolarization and depolarization with what's actually happening in the cardiac cycle. All right, we're going to stop right there.